Okay, let's learn about the university where I am working nowadays. So this is my university, guys. Beautiful auditorium we have over here. We get lot more international colleagues, even from Malaysia, from Pakistan, from US, from all around, you see it. We have a pretty sports, even car races. So we have buses that bring students from across the town, and this service is free of cost. Okay, so all happiness over here, and you are very warm welcome from the University of Southern Punjab. Anytime you like to visit this university, you are more than welcome. So now we will go to my presentation. Tarima kase. So I'm so glad. So I still remember a little Malay, not too much. Anyway, so let's start my presentation. Okay, guys, can you see my screen?
Yes.
That is good. Okay, so today I am Professor Emeritus. My name is Muhammad Mukhtar, and I am serving University of Southern Punjab. And today we are going to talk about cell biology in the era of artificial intelligence. It will be just like a story. I won't be telling you very tough things like your professors tell you. So I will make your life very easy. And this lecture is being offered to the University of Malaysia Pahang Al Sultan Abdullah UMPSA Flexible Learning in Micro Credential Center. And we have this. Okay. Here we go. So Tarima Kase to Dr. Noor Maiza Binti Sabri. So a big clap for her. Can you find out in this photograph or not?
Yes.
Okay, yes, you find it. And this is myself over there. I was a faculty member of the Faculty of Industrial Science and Technology. So I had a great time over there. Really enjoyed the great weather of Gambang that you are enjoying nowadays. So also another Tarima Kase for Dr. Noor Maiza, because she developed a very good poster of my presentation. So special thanks to her. And I just want to let you know that I came back from Malaysia to Pakistan but I keep the good relations with all my Malay colleagues. So here it is. Anybody know who he is? He is your Deputy Vice Chancellor, Dr. Ahmed Ziad Sulaiman. So he came to my office and we offered him lots of flowers. He gave a lecture over here and he is standing in my office and we are sharing good smiles over here. Anyway, so this is the general things that we shared with you about the UMPSA and University of Southern Punjab. Now we will go to our lecture and what we are going to discuss today. So there are 5 things that we will be discussing today. First is the historical aspects of cell biology. Then this cell biology, how it moved from cell biology to cytology. The cell biology evolved into cytology. We still call this science as cell biology, but basically we are living in an era of cytology. And then cytology further become like you see over here. It become like a molecular cell biology. When the human genome comes over there, and then we will be talking briefly about the translational application of cell biology, stem cells, and so on. And finally, we will talk about what is cells and AI. So this is the menu for today, and this intellectual menu. So we will be discussing about this one. And what I will do after every aspect, I will be stopping. And if there will be any question, you can ask me, and we can discuss about that. Okay, so let's talk about the historical aspect of cell biology. The first is, how many main types of cells are there? You might have learned that there are 2 types. 2 means 2, and there is a 1/3 also that, because I am a virologist, so we can never call them as a prokaryote or eukaryote, but that's a different group because they are in both categories. The virus, we call them as they are living and they are non-living. So if I ask you a question, are viruses living? You say yes or no, both answers are correct. If I ask you non-living, you say both yes or no, whichever you pick up, that will be the correct answer. Anyway, but we are very clear about the prokaryote. They do not have a nucleus. They do not have membrane-bound organelles. You can see it over here, too many organelles. And the examples are bacteria and archaea. Then we have eukaryote, like we are eukaryote, we are human. Our cells have a nucleus. Our cells have membrane-bound organelles, and we all, human being, animals, plants, fungi, they all come in the category of eukaryotic organism. Now, what is the common factor in all cells? Cells vary. Sometimes there is a neuronal cell, sometimes epithelial cell. There are almost above 200 different types of cells in a human body. The most important element or most important factor in a cell is this, what we call it. Anybody can tell me what this is?
Yeah.
I heard that DNA. Yeah. This is deoxyribonucleic acid. That is the genetic material. And this form, this RNA, and different proteins are formed. So basically, because I made my lecture very simple, I will request, if there will be any specific lecture, we will have another talk someday also. But today, I just want to make sure that how this cell biology, this science, how this has an biological or cell biology. And of course, this DNA, we call this one as a genetic material also. We call this one as hereditary material also. One thing, I won't be talking today about viruses, but you must know about this one, that viruses can have their genetic material as DNA or RNA. There are viruses whose genetic material is DNA. There are viruses whose genetic material is this RNA. But remember, all eukaryotic organism and prokaryotic organism, their genetic material is DNA. So we have to remember this one. Now, anybody know that how much time it took? Now we know about the cell biology. We know about the, this cytology, everything. It's almost 358 or 360 years ago, the story started. And some people say it was 1665, but some say it's 1667. And as of now, today, after 360 years, we are studying cell biology. We should be thankful to somebody who did a wonderful job. And who was that person? Anybody tell me? If not, yes, that was a book, that was a scientific thing published in Micrographia. That was Robert Hooke. Because what he did, he developed a crude type of microscope over here. And here is the specimen, here is the light. And, nowadays the microscope over here is eyepiece. And of course, the oculars. That was a very crude type microscope. And he looked into these Cork, and he saw that there are different parts of this Cork. So, what he wrote in his article, that is like, I could exceedingly plainly perceive it to be all perforated and porous, much like a honeycomb, but that the pores of it were not regular. These pores or cell were indeed the first microscopical pores I ever saw, and perhaps that were ever seen, for I had not met with any writer or person that has made my mention of them before this. So these pores are cells. First time in the history, the word cells was being used. So this is from where it started. And after that, after Hooke's microscope, that was the basic microscope. He was looking in the Cork. Now we know a lot of things. At that time, he was just looking some part of the Cork. Now we know all these things that has taken almost 360 years. So, all these developments, how they have taken place. Anybody can imagine about this one? So I know there is a role of different scientists, but technically it is the technology. It is the technical things that has been moving forward, And then, of course, yes, before we go into the technical things, so you might have learned about the cell theory. And cell theory, I mean, of course, there is a role of number of scientists. That's what I advise you, I guide you, that you have to work together if you want to make this world as a good place, beneficial for human beings. So that is only possible. Just, Tala, is it going okay? Okay, thank you. I just want to confirm. Okay, so there was a guy, scientist named Theodore Shevon. He studied animal cells through microscopy. And he said that these animal cells crystallize from the material between other cells, meaning when there are a lot more cells, between them crystals are formed and new cells are formed. Then Matthias Schleiden was studying plant cells, and he said cells were seeded by the nucleus and grow from there. That nucleus of the cell make more cells. But technically, they were right over here. I mean, that they were studying these animal cells. I mean, Mattia was studying these plant cells, and they were okay. That their observations were okay, but these observations were totally wrong. That cells never crystallize, cells were studied, and this, I mean, once we enter into the world of cytology, we will be talking over there that this is not the case. Rather, I mean, this was, I mean, if I was checking their paper of Theodore Shevon and Matthias Schleiden, I mean, a tough professor might have given them no, no, no. But, I mean, of course, they did. That is the way the science moves. Some scientists, like you, will be coming in the field once you will be working. You will be doing good things, and they will be good according to you. But maybe the next generation comes, the new scientist comes, they look into this one, they find out, oh my God, there is some issue, some problem. They will try to fix it, and that goes like that way. Anyway, how it get resolved? This is a guy named Rudolf Virchow. He saw cells dividing under the microscope. You know, it's a fascinating story. Once you become a researcher, I mean, you just become, I mean, crazy. Like for your science, for your research, you become very dedicated. When I saw Rudolf Virchow work, it reminded me when I was doing my postdoctoral fellowship. So I was making neurons, these human neurons. So I used to see them under the microscope for hours and hours, just watching them, that how their dendrites grow, how they come across with each other. And sometimes I do little, things. I just play with them also, put a little acid over there. So once I put the acid, all those dendrites start getting shrinking. So that used to tell me that they don't like this one. This is not good for them. So that is the reason probably Rudolf Virchow, when he was studying cells under the microscope, he saw these cells are dividing. So that gives the idea of cell theory. And there are three components of cell theory: that all cells come from existing cells, all organisms are made up of one or more cells, all the life within every I'm sorry. Is it okay now? Thank you. Thank you very much. Thank you. So, I mean, so that's what is going on. So we call this one as a cell theory, and then all these advances, be thankful to these microscope. I mean, of course, the first microscope that was developed by Robert Hooke, and of course, there is another guy named Tony Leeuwenhoek. I mean, that comes after Robert. He saw these living cells under the microscope. But I mean, the real story comes once this transmission electron microscope was developed. It start giving cellular detail, then become this phase contrast microscope. It gives more resolution of cellular structure. And in 1957, so I mean, it is just 1 year before I was born, this confocal laser microscope was developed, and this microscope led to, like, I mean, we were able to know about the cellular organelles. And after that, talk about 1984, this cryo-electron microscopy, this three-dimensional view of organelles and molecules. So this is the first part, that how this cell biology start developing in earlier days. And we covered almost 360 years, and we are over here on the this cell biology, and this all the information that has come over here, that has led to the idea of cytology. Okay, the science of cytology came into existence. Okay, anybody has a question so far?
Questions?
If not, okay, because I am just giving basic things. If not, we can move further. Then I will ask everybody question. Whosoever is sitting in the class, either ask me question, I will ask you question. Okay, that's good. It looks like, so I mean, because this was very basic. So let me complete the second one. And if nobody asks question, then I will put a fine on everybody with one question. Those who are sitting over here. Okay, let's go to this. This was these different types of microscopes. They have given us the details of cell structure. These cells, we all are made up of these cells. The basic unit is cell. You know about this one. We all human being, we start our life with cells. One cell comes from the mommy, one cell comes from the daddy. These two cells unite, and the life comes into, like, we go with this one. And every cell that is coming from mommy has the complete information about that, how we will be, what will be the hair color, and all these things. Same is from the daddy, and that is the reason that human beings are called as diploid also. So we have two copies of everything. If one copy get bad, the second copy will keep working. Anyway, someday we will talk about that one also. So now the cell theory, of course, it was all cell biology. Now cell biology turned to cytology. And what cytology basically is, we will be talking over here. Cytology basically helps you to know that all these and microscope focus that within cells, there can be like know that cells grow apart. And then the end of cell there. You might have heard about this one. So this was an era of cytology in which scientists studied cell structure, how cell function. Right now, you are listening to me and we are communicating. You know, your in mind cells, like every cell is doing different job. Like I am moving my hand, so there are different muscle cells, different proteins, different things going on. So if you are not moving hand, you are sitting quiet, you are listening to me, your neuronal cells, they are trying to, I mean, your eyes looking on the lecture. So they are trying to perceive, they are trying to understand that what is being discussed over here. So that is basically every cell has different function. If there is a neuronal cell that is basically trying to learn, remember things and everything, if there is a epithelial cells, your skin cells, they are trying to protect you, make you pretty, make you, I mean, all these things that keep happening. Then these cells have their behavior also. You know, once we are young, like in your age, everybody looks like, I mean, a pretty skin, everything good. But once people get old, then you know, you start getting these different, like the behavior, it's just change in the behavior of cells that I am looking old. Okay. Otherwise, it's the same cells that I used to have once I was in your age. So that's what it is. And of course, the most important thing is cell division. Hey, here we go. They keep dividing 1 to 2, 2 to 4, and things like this one. And of course, you know, we know about immunology and thing like this one. We won't be talking today about that one. But this is like this was the era of cytology, the study of cell structure, the study of cell function, the study of cell behavior, and the study of cell division. Okay, so not out of cell biology, but world of psychology and psychology. Okay, how about now? Okay, I think probably somewhere it goes off. Thank you. Thank you. Okay, so this is advanced cell biology. Now we know the nucleus is the genetic control center. It has all the information that parents provide to their sons and daughters. Okay? And mitochondria is the production of energy. It's everything. And you know that energy is in the form of ATP or adenosine triphosphate. And then we have a ribosome. They are synthesizing protein. We have endoplasmic reticulum. They get involved in protein and lipid processing. We have Golgi apparatus, packaging and transport, cell membrane. So it's quite possible that you have studied all these things. My job is to link all these things and to let you know, to appreciate that how cell biology has been evolving. I know Dr. Noor Maiza is a great teacher. She might have taught you a lot more good things, but I am just trying to collect all these things and show you a picture that how this world is moving in a good way. Okay, so what is benefits of cell biology? There are a lot more benefit of cell biology because in cell biology we can see the structural detail. The cell biology has helped us in understanding different types of cancer because the cells are taken, they are watched under the microscope. That is the way the people learn. So, of course, this is the branch of cytology that we are talking over here. It helps us in understanding all these things. Cell biology has been helping us in regenerative medicine. This stem cell therapy. There are both approved and unapproved medicine. They are going on in a big way. Like people can look younger. People can this is being used. Those people who get these rheumatic disorder, joint pain, all these things. So this is being used a lot. And of course, cancer and stem cell research. And I will share with you a fascinating story of a baby that is surviving. That baby was born last year. And he was just, his parents were thinking, doctors were thinking, they did not even give a name to that baby. They were thinking that this baby is not going to live anymore. So we will be sharing the story of that baby also. That baby is this top 10 story of the science of year 2025. And all the credits go to cell biology and cell molecular biology that we will be talking shortly. Okay, so here it is. Now I just will take few minutes and I will share with you my story because I served the US government for 20 years. And of course, I served the University of Malaysia, Pahang also. And there are a number of other universities in the world where I had opportunity to serve there. So, but my 20 years in US were just to work on the cell biology of brain. You see, our brain is covered with a structure that we call as a blood-brain barrier. So this was my job, that I have to collect cells that form the human blood-brain barrier and develop a model of, like a test tube baby, develop a model of brain in a test tube. That was my job. And it involves like millions and millions of dollars because we were getting cells in ringgit. It could be a lot more money. Okay. So what we developed, my laboratory developed a model of human blood-brain barrier that was a typical experiment of cell biology, like you take the cells and you make the organ from them. Okay, because a lot more cells get together, they form the tissues, and then these tissue form the organs. So that's the way life goes on. So, there was a heavy funding for this one. Now, what is the benefit of this blood-brain barrier? It protects your brain. Like it does not, once we eat food, all the nutrients, this goes across the body. But in the brain, only brain like to have the good things. This blood-brain barrier, this allows only good food to get in and everything else that get out. And you might have heard the word brain hemorrhage. So anytime that creates the brain hemorrhage, and people hardly survive from them. So very few people survive from there. It is this blood-brain barrier. So it is like these cells, they get together with each other, and they one cell with other, they just join with each other. They form what we call them as tight junctions. Means these tight junction make sure that nothing goes inside the brain, only good thing, only yummy food, the one which brain likes it. Why we were developing this blood-brain barrier, I just want to share with you, because we want to know, because people get this AIDS, HIV infection, and this virus goes into the brain and hide over there. You treat the person, you can make the virus zero in the body, and once you withdraw the medicine, the person again gets sick, because the virus that was hiding in the brain, that comes back. Basically, that was the idea from where we started, and we were working on this. Anyway, so this is again this diagram of this brain and thing like this one. And here we go. So now there were lot more cell biology experiments you might be doing also, or you might have done, or in the coming days you will be learning about this one. Now the techniques might have evolved in a great way. So one of the thing was that my cells, the cells that form the blood-brain barrier, they are brain endothelial cell and astrocytes. They might be expressing one of the marker that we call a von Willebrand factor. Bit tough science, but you should understand this one, that there are certain feature, certain characteristic of cells, and then these different proteins, ZO1, zonacluda 1, zonacluda 2, weak, adherent, CD19. These are different scientific names. You don't need to remember. But these cells, if they express all these one, only then they can form a good blood-brain barrier. If they are not expressing, people get neurological disorder, people get different infection. You know about this one. So that is the reason, that is the problem that we see over here, that once the blood-brain barrier is not good, people get, so I established that model, and I won't be talking in detail of this one, but I can definitely share that one. And we studied this one, and this was funded by a company that is a Pfizer pharmaceutical. This is one of the biggest pharma company, and we published something almost long time ago. But research is always going on this thing. It's the similar thing. Okay, so this was basically the model. As you see it, we try to develop the blood-brain barrier model over here. We used to put the virus over here and then find out whether virus can get to the brain or not. And then we studied the molecular cell biology, that what goes on over here. This is a bit complex. It will get boring. So we return to our basic things, like what we call as, so now we have covered this historical aspect of cell biology. And we have covered cell biology to cytology, and from cytology to molecular cell biology. So now we have to go over there And I would like to, if nobody is going to ask me question, should I ask the question? Okay, guys, how many chromosomes you all have? Dr. Noor Maza, how many chromosomes? 46. Wow, that's what it is. That's what I want to know. And where the chromosomes are located? Where they are? Nucleus. Wow, we know a lot about cell biology. That is fascinating. Like these chromosomes, they are in the nucleus. Okay, so how many boys are sitting over here? Is some boy sitting over here? Okay, boys, tell me what is the specific type of chromosome pair you have it? XY and XY. How about girls? XX. Wow, you made my day. I know this is like, I think I don't need to teach you this, discuss. But anyway, we will discuss a brief about this molecular cell biology. Okay, let's go with that. Okay, here it is. Now, we started from a microscope. Microscope help us in cell biology. And these advancements in microscope led us to know the cytology that we learned about this one. And now we come into the fascinating world of molecular cell biology. Okay? Molecular cell biology, it's all hangs over here. Though there is a mitochondrial genomics also, but main molecular cell biology is over here. They are the chromosomes. As we know about this one, ladies, girls have 2X chromosomes, men have one X and one Y chromosome. Okay, so that's the way the generation keep going on. So if one X chromosomes match with the other X, so one X chromosome from father, one X chromosome from mother, so that will be a baby girl. One X chromosome from mother, and from the father, if Y chromosome is coming, then there will be a baby boy. So that is the way life moves. Anyway, so now we talk about that how these chromosomes moves forward. Okay, so here it is. There was a project. You might have, I don't know, there was a human genome project that we observed this one. Like people of my age, they are the lucky one. They saw very crude cell biology to the advanced one. But you are entering into the advanced cell biology and you will be enjoying more, hopefully, this new type of sciences. Once they come in, you will be learning more and more. So this human genome project, that sequence all the 46 chromosomes, 23 pairs that you told me, and there were and this, every cell has the complete genetic information. Every cell has this 3 billion base pairs of human DNA, and there are almost 20,000 to 21,000 genes, you know it. The human genome project that transform cell biology into molecular cell biology. That's what comes over here. And what has been happening in the molecular cell biology, now we know that how many like genes are there. I mean, you might be familiar with this one on what chromosomes, like these 23 pairs. I mean, if there is some disease, which chromosome is the problem for that one. So this has given us an opportunity to study that one. Okay, so and then now we know it that these chromosomes like they are involved this our DNA, like as I was telling you that why my skin cells look age, why your skin cells looks young, because they are involved in the aging also. There are changes in these chromosomes that basically come up in a way that the older people looks older. So we are talking about, cells. Okay. And there is like I have 46 chromosomes, you have 46 chromosomes, but my chromosomes are different than yours because I am, and they are telling me that I am an old man, and they are telling you that you are young boys and girls. Okay. Then there is like, I mean, what is the effect of diet and all these? This is a fascinating story. One thing I have to tell you, I have to advise you, never learn science that I have to remember this. Learn science as a story. If you learn science as a story, then you will enjoy more, and that will be a fascination thing for you. Now, this Human Genome Project, this told us everything over here, like how we age, what is the effect of drugs and pharmaceutical on cells, what is the effect of environment, like it's cold outside, what is going on in my cells. So all these things keep moving over here. Okay, and then it also told us the molecular basis of disease. Like, I mean, there were inherited disorder, and now we know at molecular level, like, if you have taken any course about this one, okay, otherwise, I don't want to make your life more complex. Like, there are certain genetic disorders. Mommy has that genetic disorder, can be transferred to baby, but I mean, once these genetic disorder, like some of them they are dominant, some of them they are recessive, we won't be talking in detail about this one, but all that information that we accumulated, they were due to, like, I mean, this molecular cell biology that made our life so fascinating. Okay, so the most important, the most fascinating thing is personalized medicine. Okay, personalized medicine, or like we all human being, we are unique. Allah subhanahu wa ta'ala, God has created us in a unique way. You all are sitting together. You all have some genetic differences. They are very minor differences, but they make us like a human being. And of course, due to these differences, so like, I mean, the medicine, some medicine are good for certain people, but they are bad for others. So, I mean, one of the good example is one of the genes that is a cancer that is among ladies, and same medicine can cure people with a particular symptom a lady is having, and the same medicine to the other due to genetic difference can be very bad, can be very create problem for that person also. So this was the era of personalized medicine, and that based on genetic changes in the cell, in the science that we come across with this one, that was the molecular cell biology. Now, you might have heard about this one, CRISPR-Cas9 story. Like, if there are changes in the genetics, if there are changes in the chromosome, in the DNA, that can be corrected by a CRISPR-Cas9 story. And there is a lady who got the Nobel Prize also, Jennifer Doudna. It was developed in her lab in California, and that technique is now being used to treat humans. And here it is. This is one of the, just give me 1 second, and you look in this slide, and we will be talking about this. Okay, I'm back over here. Anyone has, my question is to students, have studied this story? Yes, no? No, okay. This is a fascinating story. And this story is about this little handsome blue-eyed baby. Okay, this baby was born in August 2024, and he was having a deficiency or a gene defective. That gene is carbamyl phosphate synthetase 1. This is inside the liver cells. This baby was brought to the doctors because I served in Philadelphia, and this baby was treated in a hospital that is a Children's Hospital of Philadelphia. Philadelphia is one of the city in USA. It's close to New Jersey and New York. You might have heard about New York and Washington. It's in the middle. And that's a very, that used to be the first capital of United States also. I lived over there for 20 years, and my son and daughter still live over there. So I have a house over there. So we also, I remember when my son and daughter get, sick, we used to take them over there. So that's one of the very good hospital. So over there, once this baby was brought over there, so the doctors, they did not give any name to this baby because they were thinking this baby will die. This baby won't be able to survive. So, and they gave this baby a name, Patient Ita. Okay. And parents, mom and dad always, they love their son and daughter, but they gave name to the baby as KG Muldoon. Okay, so the baby was brought over there, and the disease was this one. Like in the liver cells, this carbamyl phosphate synthetase gene was defective. You see, this is a typical example of cell molecular biology. What it does, that inside the body, that ammonia and that carbon dioxide that is being formed, it transforms into carbamyl phosphate. Okay, so now this is a very bold experiment. There is, as I shared with you, there is a technique CRISPR-Cas9. Through that, one can correct the changes, one can make changes inside the cells. So once these cells divide further, they will be having those changes over there. Okay, so now this either you call it, I mean, he is no longer patient Eta. Now he is a KG Muldoon because he is surviving. And how old he is now? August 24, 1 year, and from almost 1.5 years. These are the two doctors, Kiran Masonoro and this Rebecca Ahrens Nicholas. So they basically use this CRISPR-Cas9 methodology to correct the gene that was defective in this baby, and that gene was this carbamyl phosphate synthetase. They change it with the correct version of the gene, and fortunately, this baby is like with the parents, is surviving. This is the fascinating story of molecular cell biology, like how we started from a cell biology, we move toward the molecular cell biology, we move toward the cytology, and now that cell molecular biology is helping that people, like, I mean, those babies who don't have any, like, I mean, people think they cannot live longer, now they are surviving. So this is like a fascinating translational application of the science of cell biology. And imagine you are studying this fascinating course, and that you can make, if you want to pursue your career in future days in cell biology. So these are the fascinating stories that need to be repeated again and again. And this question, my question is, how can this help more children like KJ? So imagine now the life of one child was saved, but this disease happened across the world. This disease happened across the globe. So how the science can move forward and the people life can be saved. So you know, we started our journey, our story started from a very basic thing that was like this Hooke's microscope, and we have been traveling through the cells. It took us 400 years. Now we know everything, and this year 2025 is seeing that how this cell biology is saving these blue-eyed handsome boy. So this is the way, but now it lies with us. It's our obligations that we should learn this methodology. And there is an article, Nature 10. So it's worth reading. I mean, it's freely available on the net. So, I mean, this is one of the story of 10 stories. Okay, so, I mean, there is another way. Now there are a number of medicine that are coming. So you might have heard about this is a disease, sickle cell disease. I mean, this is one of the genetic disorder in which on chromosome number 11, the hemoglobin is not good. So either the same technique that we just discussed that could be used, or there is an other technique that has been developed, and this is one of the medicine that is recognized by the United States Food and Drug Administration. Its name is Casgevy. This drug was approved in 2025 in this year. So, because we are discussing about our journey over all these years, now if somebody is having this disease, this sickle cell disease, what it is, these are our normal red blood cells. If there is this change in the hemoglobin, so these cells become like a state. And once they are flowing in the blood vessel, they just stop the blood vessel, and the patient feels very painful condition, and they are like, almost very challenging. Okay. So, this gas heavy is basically, cell therapy. So, this is the that we have been discussing today is a technology like from a patient, the stem cells are taken out, and these stem cells, you know it, because they have the gene for these sickle cells, due to which the patient feel lot more painful condition, and these stem cells, they are corrected with normal gene for hemoglobin. Okay, like this one, the gene that makes this normal hemoglobin, and after that they are reinfused in the patient. This is a very complex clinical protocol, and of course there is a clinical procedure involved over here that I don't want to. We don't have any time because it's a but I just want to make you aware about this one, that this is the way the life moves over here. Okay, so and there are almost approved cellular and gene therapy products 48. So these are different companies, and you can go to US Food and Drug Administration site, and you can learn more. It's fascinating to watch that how the people are being treated for different diseases. All credit goes to this cell biology and its application, its advanced application, which are making these fascinating entry into the world of therapy for human being. Like patient Eta can get treated and the boy survive, and the same is for as I shared with you the example of sickle cell disease, and there are a lot more other disorder for which these medicine have been developed. Okay, so we almost cover historical aspects of cell biology. We covered cell biology to cytology. We covered cytology to molecular cell biology, and then we have translational application of cell biology, stem cell, and so on. So, the last thing in the 5 menu course that is left over, that is a cell and AI. Okay, so let's briefly talk about this one. That what this one is. I just have a question for you. How many of you are using Chat GPT?
Yes, yes.
Everybody use Chat GPT. That is fascinating. But now the science is moving so much. Like, I am an old man, almost 68-year-old, but I love to learn like you learn. So, I mean, there is a something called agentic AI. That basically is like you can program and you can have something that keep working for you. Like you want to get information that what is going on in Kuantan. So that agent, you create an agent, and that will keep telling you that this thing is going on, but anything comes on the net that you can get from there. But I almost spend like 5 to 6 months. Any video, any course comes in, I took those one. And one day I was on the chat GPT because I have a version that I subscribe, I pay for that one. And it was fascinating to note that this chat GPT has the power of agentic AI. So everything that I was learning in the past 6 months, that becomes zero. So this is the way now science is moving, and same is going to happen in the world of cell biology also. So, what is being envisioned, what the scientists are thinking nowadays, like they want to develop AIVC. What AIVC is, that it's artificial intelligence virtual cell. It's a fascinating concept. They want to, scientists are working on this one. To me, being a neurobiologist, it will be challenging for the scientists. But once there is a will, there is a way. So they are working on this one. They want to develop this chat GPT like model of a cell, and in that cell they want to observe like how protein synthesis is going on, how cell is dividing, how if there is a change that goes on. So everybody they want to do on the computer. This concept is good like gold. There's a lot more money being spent on this one, but it will take some time. But definitely, we will see it in our lives. Like they will be able to get this thing done. So, the idea is to develop a computational model that simulate the biological function and interactions of cell. So what it will be, it will be a multi-scale, multi-modal, large neural network-based model that can represent and simulate the behavior of molecules, cells, and tissue across diverse state. Uh, I mean, in the past, the things that we don't believe that has happened. But if we look into the personalized nature of human being, if we look into the diversity of cell, so I know they might be able to develop something, let's say for a neuronal cell, but you never know that they will be able to develop for each and every cell. But in cell biology, maybe Dr. Noor Maiza might be developing a course like AI cell biology in the coming days and molecular biology. So that will be the fascinating. We will see these things in our lives. So, and the next one is what is needed over here. So this is what you need if you want to develop a artificial intelligence virtual self. First thing is, as I discussed with you, there are normal cell, there are tumor cell, there are immune cell, there are nerve cell, E. coli, yeast, prokaryotes, eukaryotes. So there are too many organisms. It will be a story like initially there was a human genome project, and now there are projects for everything. But I mean, being a scientist, this is a very good paper in Nature. I took this photograph from there. If we are determined to put artificial intelligence in our life, so definitely what is needed, like there is a lot more knowledge that is needed, cell biology, cytology, molecular cell biology, and of course that what we have been discussing. And then there will be a static system, the images for all these cells, molecular structure. Last year there was a Nobel Prize even to AlphaFold. Like in a cell, there are thousands and thousands of molecules being synthesized and reaction ongoing. And then if you want to make it in a and then dynamic state, like you better know about the proteomics, genomics, and lot more thing, and that will come the world of this what we are envisioning, AIVC. So this is like a challenging environment that has to come in future. So I know there are few minutes left over. So I just have to close my lecture over here, and to everybody for listening to me and for coming in the class, and particularly to your professor, Karima Kase. So I will stop over here. So stop sharing, and thank you very much. Thank you for listening. So if there are any questions, so I would love to listen your question. And okay, there is a question. I see hand over there.
Okay. Good morning, Dr.
Selamat pagi. Good morning. Yes.
As the story from KJ just now, they're using the CRIPS therapy to treat the baby. So, is it the CRIPS therapy can treat the syndrome down?
Dr. Nurmaza, can you repeat this question for me, please? Sorry, I mean because my this recorder is not so good. Yes, please.
Okay, so basically, she's asking about this CRISPR technology.
Yeah.
Genetic. So can it be used to treat Down syndrome?
Yes, that's a very great question. Thank you very much. Down syndrome is also a genetic disorder. It's inherited one. You know it. Like definitely we will be seeing that one in the coming days. All genetic disorder, they can be treated. I mean, that's what I want to get from you. So you name it, you identify the disease, and you can definitely treat this one. Karima Kase. So this is a good one.
Okay. Question? Okay. I know what is the challenges of studying the blood-brain barrier.
Okay.
He wants to know the challenges of studying blood-brain barrier.
Blood-brain barrier?
Yeah, the challenges to study it.
Oh, it's a tough one. Don't ask me. It's a very tough. I spent my 20 years, and that was because, every time I cultured these, I used to isolate human cells and culture them, and I and even my technician They come, they say the cells are contaminated because the brain cells are yummy for the microbes. The first challenge is like, this is a very smart question and this is a very intellectual question. The first is if you are working on in vitro blood-brain barrier model, it's a hard to culture them and you need specific expertise. The next one is if you want to study blood-brain barrier of living organism, then there are ethical reasons. So, the organism gets sick. There are a lot more challenges. There are, you are very right, but somebody has to work on this one. Both for in vitro, for in vivo models, these are the prominent challenges. In vitro, how to culture these cells in an effective way. In vivo, nobody want it. Like if I want to study human blood-brain barrier, nobody will let me put the electrodes and everything over there. So that's the reason, there are challenges over there.
So maybe she can come to you and be your successor.
Yeah, I know. I see a lot more people. They will be studying this one. So I know they are very, University of Malaysia, Pahang is the good one. So you all should work on this one because there is a lot more opportunities on that side.
Yeah, applications.
Regarding your blood-borne pain, regarding your blood-brain barrier study, have you published any papers that you can refer to? Because I'm quite interested in your research.
Oh yeah, I have almost 50 papers. You can find not done. So these are all the challenges, So just write my name, Muhammad Mukhtar, on the PubMed. That's a very good one. Guys, if you are interested in science, so I know we should be thankful to US government that they have a database that is National Center for Biotechnology Information, NCBI. So it's a www.ncbi.nlm.nih.gov, and you go over there and you can do author search. But I can definitely send the first paper to your professor and she can share it with you.
Okay. Any other questions? Okay,
Good morning, sir. I have a question regarding the blood-brain barrier also. It's very interesting. So, when there is a damage or a degeneration due to old age, is there a medicinal or natural way to recover or improve the blood-brain barrier cells?
It's a tough one. I may not be able to answer that one. So, but definitely this is the question that science is asking. That once the people get age, but I can share with you one thing, like these forces, army forces, once they go in the war zone, there are blasts. Due to that blast, their blood-brain barrier get leaky for a small time. And that is the reason these army personnel, once they go back into the normal life, they have a psychological problem. So, this is an area that is under discovery. Of course, with aging, there will be a lot more effect on every cell, as we discussed about this one. With aging, there is a changes in the chromosome and everything. So definitely there will be changes in the endothelial cells chromosomes also, and that get problem. And the medicine, if you ask me, there is a regenerative medicine. That is a new branch that is coming up. You can put the cells which or new one. Like I am 68-year-old, why I cannot look like a 22-year-old person? So that is the future that is coming up, and you guys are going to work on that.
Okay, I think we have time for only one more question. Okay.
Good morning, doctor. I want to ask, can we use also the concept like AIVC to predict also the mutation of virus?
I think your question was about the viruses. Is that right?
Using AI to predict the mutation of virus.
Basically, I am a virologist, and I was just studying this blood-brain barrier in respect to viruses. That how these viruses get into the brain and how these neurological disorder, due to virus comes up over there. If I understand your question right, I think that is the reason I developed that model and I was using over there.
All right, so I think there are more questions they want to ask, but we don't really have time right now. So maybe if they have any question, I can give it to you through emails or WhatsApp later.
Sure. I will send my presentation. I will send you blood-brain barrier. Give it to that. Everybody. Sure.
I'm very interested in your presentation. I think they have asked more questions to you than all my lecture combined.
That's a good one. We will have another lecture someday about the things they like to learn.
Yeah. And hopefully, after this, we can collaborate to more things like sharing courses and so on. Okay, that's.
Okay, terima kasih. Thank you. Sama-sama. Thank you. Terima kasih.
Yeah, Assalamualaikum.
Waalaikumsalam. Have a great day. Enjoy your day in Gembang. I miss it. That's a pretty town. Bye-bye.
Yeah.
Yeah.
Okay.
Thank you.